It's all about sexual reproduction. We're going to have two haploid gametes coming together to unite to fuse to form a diploid zygote. Now, the way this works in humans, and I just want to put this broadly out there, the male's producing sperm, the female's producing eggs, the sperm and the eggs come together. It's not just magic, it doesn't just happen like randomly.
Happen in your sleep.
Right. And you form a diploid zygote. The reason I have to explain that is because I didn't realize it, but apparently there's some people out there at the college level who really didn't know. I had to, I said, guys, all right, so sexual reproduction, you all know how it works. We're going on, and they're like, egg and sperm, and I was like, yeah, they were homeschooled. Their parents were really, no, I wouldn't say conservative. They were just very reluctant and embarrassed to talk about the way sex works.
Okay.
It's okay, my parents were too.
Yeah. See, the town where I grew up had a very, very bad health program.
So, the way this works, haploid, they fuse. This becomes known as a zygote. That zygote then is going to very quickly undergo mitotic cell division. Okay. As you are developing, I'll just put this up here because it's not in the notes, but you know, I'll do it anyway. You've got a zygote. That zygote's going to split in half to create two 2N cells. Those split in half to create four 2N cells. Those split in half, and how many have you produced? Keep going. 8. 8 go to 16. 16 to 32. 32 to 64. 64 to 128. 128 to 256. 256 to 512. 512 to 1024. And so on. Eventually, you have enough cells to go from the single zygote to what's known as a blastula, which is a hollow ball of cells. Blastula. Not really. It's got a different root. That blastula, this hollow ball, is going to what's called invaginate. It gets an indentation in it. It's almost like taking a balloon and shoving your fist into it. It forms a concavity around your fist. That blastula then has undergone gastrulation and become a gastrula. This is where you get your skin, your digestive system, and all of your internal organs. That's development. In order to get there, though, you needed to have an egg and a sperm. So we get those egg and those sperm through this process called meiosis. So to produce these gametes, we have to first go through G1, S, and G2. Where have we heard of this before? You're right, it's mitosis. It's pretty much the same stuff. There are a few slight differences. In mitosis, during prophase, you get your homologous pairs have formed and they've thickened. During meiosis, you take those sister chromatids and those homologous pairs and they come together to form a tetrad. So instead of having, time for a puppet show, you've got chromosomes. So these chromosomes look a lot alike. During S phase in mitosis, they replicate. So now you have two sister chromatids joined together at a centromere. Does that make sense? They're going to line up at the center of the cell during metaphase and then split apart. In meiosis, it's slightly different because what happens is you start off with two chromosomes. They undergo synthesis. They copy each other. But instead of staying apart, they come together to form a 4-chromatid module called a tetrad. Or synapsid, if you want. Those synapsids, those tetrads, allow us to undergo a process called crossing over. Crossing over is where there's a physical exchange of chromosomal elements. Here you've got the tetrad formed. These legs are so close together and so chemically similar, when they cross, they can actually switch spots. So you've got your dad's is red, your mom's is green. They're able to swap pieces.
And now you're part of your father, part?
Now you're mixed up, yeah. It's called a recombinant chromosome. Or recombinant chromosome. They've recombined.
Oh, okay.
It's the equivalent. You guys ever run a three-legged race?
Yeah.
And there's a few ways of doing that. One is, you tie yourself to another person's leg and you sort of like run along together. But what inevitably happens is one person just picks up the other and drags them because you have that one ultra-competitive person.
Yeah.
Yeah, okay. As long as we're there. Slow down, slow down. Oh, my leg. It'd be like at the end of the three-legged race if your leg and their leg switch places. Yeah, they're similar enough. You got a knee, they got a knee. You got ankles, they've got ankles. You undo the tie, and suddenly you're wearing their shoes.
Then you got two left.
Yeah, well, thanks for pointing out the flaw. Cool. Where these things cross over, where these sister chromatids cross over, you have what's termed a chiasma. Right, because it's named after the Greek letter chi. Looks like an X. The arms of one chromosome cross over the other. So here you've got A, B, C, D, E, F, G, H, I, J, K, L. Here, same thing. Here, lowercase, here, lowercase. Where they cross, now you're going to get a recombination event, a crossing over. So you have the first one stays the same. The second one becomes A, B, C, D, E, F, G, H, I, little j, little k, little l. Then little a, little b, little c, little d, little e, little f, little g, little h, little I, j, k, l. This is where we get variation. This is what allows us to be different from one generation to the next. Regulation of crossing over is highly, highly regulated. In humans, it only really happens about once, or maybe twice in a gamete. In plants, it could happen like 20 times. Plants use this as a major way that they look different from each other. So here we've got chromosome ACB, little a, little c, little b. There's a double crossover event, so we get big A, little c, big B, little a, big C, little b. Slight differences. So that when each of these goes into their own gamete, you've got one gamete that's got chromosome ACB, one that has A, little c, B, little a, big C, B, little a, little c, little b. All unique. So in order to get these unique chromosomes that will go on to make unique people, you need to undergo meiosis. There are two parts to meiosis. Meiosis 1 and meiosis 2. Meiosis 1 is composed of prophase 1, metaphase 1, anaphase 1, and telophase 1. All of which... Pardon? Yes, phases of meiosis. First phase, prophase 1. The chromosomes thicken, the tetrads form, crossing over is occurring. The nuclear envelope dissolves, the mitotic spindle forms. Metaphase. The microtubules have attached to the tetrads and pulled them to the center of the cell. Anaphase, the tetrads are pulled apart, moved to either end of the cell. And telophase, the nuclear envelope reforms, and you get two distinct daughter nuclei. So to recap, prophase 1, the nuclear envelope dissolves, the tetrads thicken, crossing over occurs. Metaphase, the microtubules push or pull the tetrads into the center of the cell. Anaphase, the tetrads get pulled apart. And then telophase, the nuclear envelope reforms to give you two distinct daughter nuclei. Question? That's the end of meiosis 1. Meiosis 2 will occur next. Do you guys feel comfortable with mitosis? Because here's the deal. Meiosis 2 is exactly like mitosis. The only difference, the huge difference, is that meiosis 2 does not have S phase. Yeah, so they'll just have G1, G2. Yeah, there's no S phase in between. If there's no S phase, what occurs during S phase? Synthesis, chromosome replication, which means we are now, by the end of it, having the amount of genetic material that we had previously. So you got meiosis 1, where you got those tetrads and crossing over happening. And then you've got meiosis 2, where you've got effectively the same thing as mitosis, except there's no S phase, no copying the chromosomes.
Where does that happen?
Where does that happen?
Yeah, where does meiosis 2 occur?
Meiosis 2 is going to occur, in the cells that are producing the gametes. So, like the spermatophyte in the plant, the oocyte or the oogonia in the plant, the primary oogonium in the female, things like that. So, not throughout the whole body.
Right.
So that's meiosis 2. You put meiosis 1 together with meiosis 2, and you end up with a haploid gamete at the end. You'll notice we start off with 2 chromosomes at the top. We end with 2 chromosomes at the bottom. The difference is these chromosomes, these final gametes are all unique. They all have different types of genetic material in them. Different, not types, combinations of genetic material. So for the inevitable question of what's the difference between mitosis and meiosis, mitosis will produce diploid daughter cells. Meiosis will produce haploid daughter cells. Mitosis produces identical daughter cells. Meiosis produces unique daughter cells. Mitosis generally produces body cells. Meiosis produces sex cells.